So welcome again, and today is day number 2 of ISCT TV 2023 in Paris. And today I'm here with ESP Ambassador Ted Kiefer because my Patrick just left me today and went to the networking
poster session.
Yeah, poster session.
He told me he was heartbroken. Okay. He really wanted to be here, but I'm doing what I can to fill his role. Hopefully I can do it semi as good as Patrick can. But
so what was your experience today? Are you having a good time?
I'm having a great time. There have been so many opportunities to learn, so many opportunities to engage, so many opportunities to enjoy myself. I mean, Paris is just a beautiful city. ISCT did a fantastic job in their selection. And then the roundtables. I mean, there's over 50 roundtables, I think, for ISCT. So the co-chairs put together some amazing sessions. One of the more thought-provoking ones was an ISPC or IPSC that, or it's like a thought challenge about IPSCs and how you can take them from development and what regulatory controls are needed up through preclinical trials, then all the way up to bioassays. So it followed the line of thought throughout the whole developed and manufacturing process and even distribution and regulatory approval. So it was very stimulating, and I'm a lab guy, so I got to think about lab assays and all that kind of stuff. So it was delicious, as food for my soul. And then the innovation sessions, seeing what people are coming up with, and the poster sessions, being able to walk through and talk to individuals about their work. I love that part of conferences. You don't get that anywhere else.
So I heard through the grapevine that you talked to somebody important today.
I
Tell
did.
us about that.
I'm talking to you.
Well, to somebody that got an award.
I did. I talked to Dr. Jan Nolta. It was fantastic to meet up with her and go through her Lifetime Career Achievement Award that ISCT gave her.
Well deserved.
Very well deserved. And I have an interview that we can cut to right now.
Okay, perfect. Let's listen to that interview.
Ça va toujours. Ted Kiefer here again today. ISCTV Day 2. We are here with Dr. Jan Nolta of UC Davis, who is our Lifetime Achievement Award winner. Can you believe it?
Yeah.
You've been in the industry since
a long time, almost 35 years.
I believe,
yes, 1987.
And ISCT hasn't even been around that long, 1992.
Right.
So what have you done to change the dynamic that got you this Career Achievement Award?
I think I was kind of one of the founding members. I remember coming to these meetings when it was just a couple hundred people, and now we have like 3,000.
A couple hundred people.
I've heard. So I think I just kept showing up.
You're the recruitment tool for all those years.
But I'm so amazed to get this award, and it means so much to me. And often awards go to people that have just really built their career through an ivory tower, and they're the big professor and everything. I'm just a girl from a small town that has worked hard, a ton of teamwork,
a ton of mentoring too.
A ton of mentoring.
You've been bringing people up all throughout your career.
Students are what make me really happy.
Yeah.
So working with young people and just helping them along in their careers. And I work with amazing teams at UC Davis Health in Sacramento. We have different disease teams working on different areas, wonderful MDs who are really committed to bringing their patients new and novel therapies, and that's what really drives our innovation and translation.
Yeah. Well, I can. This is the first time I met you, and it's unfortunate for me, but I mean, your passion definitely comes through.
I love it.
And everything that you say and you do, and the beautiful speech from Guido.
Guido Moll was my nominator, and he gave a very, very touching speech, and he said, "I'm the American dream," because I grew up, had a single mom, food insecure, all that, and got a job as a technician with Don Cohn, who's like the giant in the gene therapy industry now. But back then, Don was a fellow coming to work at Children's Hospital. I was his first technician, and then I was his first graduate student. And then we worked together for 15 years to develop hematopoietic stem cell gene therapy techniques, and then started clinical trials for babies with ADA deficiency, bubble babies. And they kept going with that for the past 30 years, and they've now functionally cured 50 kids with the techniques that we developed initially.
And ADA also, something that was terrifying for a long time, without a doubt.
kids have no immune system. They have to be completely kept away from any germs.
Yeah. We all remember Bubble Boy, David.
Yeah, the Bubble Boy.
You're changing lives throughout that. And you just defended your thesis as well, correct?
Oh, no, I am done.
Okay. Finished it?
No, right. One of my students just did.
Student just defended the thesis. Okay, that was a misunderstanding.
That was Casey. That's okay.
How did she do? And again, another student that you mentored. She's here. You have seen her with the poster.
Yeah.
Fantastic.
So I finished mine in 1994, if you can believe it, with Don. So I was his first grad student, and we kind of went through grad school together. He helped me. I helped him learn how to have students, and all good.
Yeah, it's lovely.
But that's been a few years. I'm glad to be here. It's been a wonderful meeting. It's so great to be in beautiful Paris and just see everybody in person after years of just the Zoom screens, and it's just great to be here. I love it.
And taking on new students, engaging throughout the ESP program. So it's been fantastic to have you.
I'm especially fond of mentoring first-gen students. Sometimes we think, I don't know, like the professors are these big scary people or something, but shouldn't be that way.
Never got that impression from anybody I've talked to about you.
Not from me, but sometimes students are afraid, especially if we're first gen and we come from humble backgrounds. It can be intimidating to meet the big people that have published so much, but take the career by the horns and go for it, students.
Yeah, most definitely.
Thank you.
Thanks
so much.
Really appreciate it. ISCT, stay tuned.
So hello, and here we are again, and we have our president, Jack, with us today. So welcome, Jack. Tell us, what are the highlights of this day today for you?
The highlights of the day?
Yes.
I'll speak about yesterday because it was a busy day for me, especially because we had La Présidentielle going on. A plenary theme that I'd like to continue, using La Présidentielle as a legacy term afterwards. It was terrific. Fantastic speakers. The room was packed. Even the overflow room was used up.
Wow.
And we had incredible speakers, again, speaking to the vibrancy of the ISCT, that we have highly international sought-after speakers within our family that we never get to hear what they have to say. And here, this was the right time.
Yeah.
And it was sensational. What a great plenary. So it was terrific. There's the plenary, but something that's a true delight for me as president is also to give the prizes of the society.
Oh, yeah.
And the meaning of those prizes, because there's some of them that are really bespoke to the ISCT, but I'd like to pivot, for example, to the Lifetime Achievement Award. And to me, that was that. A lot of societies like to, invite their Nobel Prize winner, and it's the 48th honorary PhD and whatever, and they're tired about it. They just come in and come out and whatever. Yeah. But here we had a homegrown, somebody went up through the ranks and made a huge difference that has, at the root of some gene therapies that have cured people, like the ADA deficiency, as part of her work, leading this incredible effort in California, and who's such a wonderful person. So Jeanne Nolta, I'm speaking of course.
I actually interviewed her. Heart of gold. In and out.
Mentor.
Yeah.
Many people.
But the thing is she's such a wonderful example of what our society is all about. Mm-hmm. So there's that. There's of course the mentor of the year. There's we had a couple of inaugural prizes.
Yeah.
For early stage professionals, for emerging economies. That's terrific. And I do want to dwell also on one last prize. I'm talking too much, but anyway, I'll carry on. I have the microphone. So I wanted to speak to, of course, the Luc Saint-Sébé Prize.
Mm-hmm. Yep.
Luc was a very dear and sweet friend of mine, and he was such a happy warrior. Luc was a happy warrior. In France, happiness is, it comes and goes. But he was a happy warrior, and he was the cat herder in chief. And again, we're talking with the French and the Europeans, and he brought them together, and he was able to boldly go where no person had gone before to make things happen for cell therapeutics. And it's a prize, I say it's bespoke, because it's not one that's, it's not for the most cited paper. It's not, it's a little bit different. It was about those that make a difference in advancing translational science in our space.
Okay.
So it was really nice. And of course, Michael May's Canadian. Who could, talk badly about Canadians. So Mike is a great guy. So that was a lot of fun yesterday. And of course, the later on yesterday and later on today, something I really, really enjoy. It's like eating French chocolates is the abstract sessions.
Oh yeah, without a doubt.
Walking around in the abstracts, and you walk up to, there's this person's obviously they're a PhD and they're there from the poster, they're waiting for somebody, and you walk up and you ask a couple of questions, and you see their eyes light up and this and that and this. Love it, and I learned stuff.
Yeah.
I learned stuff, and it's great interacting with, because they're the future. And you know what? That's one of the great things. When everybody at a conference is as old as I am, you're in trouble. And, you have all these ESPs and trainees and junior faculty members all there. I know the wind is in our sails. This is a great time to be part of the ISCT, and I think others are going to be jealous. I'll stop here, because I know you have other questions, but I can't read.
No, thank you for celebrating individuals within ISCT at every level. I mean, I'm an ESP. I've felt so welcome since I came on board, and then I had the opportunity to talk with Jan. She's been in the industry for over 30 years, and she helped facilitate the building of organizations like this. So thank you for continuing to foster and develop and getting excited about all of it. It really shows a lot. So.
Thank you, Jax, for joining us. So up next, we have an exclusive interview from ESP Ambassador Alicia Disbury, featuring Matthias Van Bolk, Innovation and Services Manager at Biotech, to discuss their work in spatial biology. So see you then.
We are here with Matthias from Biotechnique, and we're going to talk today about some of the amazing innovations this company are pushing through, and in particular we're going to talk about spatial biology. So we were having a really great conversation just before about some of the intricacies of spatial biology, but why don't you explain it to us a little bit?
Well, typically spatial biology nowadays is about the study of, let's say, biomolecules in most of the time cells or sections, 2D sections, but also in 3D possibly, where these biomolecules are studied in relation to each other on, as it says, in a spatial relations. So this is really something that nowadays is a hot field actually. Why? Because we have had about more than a decade of sequencing, single cell sequencing, and all that sequencing data can now amount to really spatially resolved data that actually broadens the scope of our biology understanding.
Absolutely. And so, like you just said, this field is very rapidly evolving.
Yeah.
So where do you think it's heading, and what role do you think Biotechnique are going to play in that?
Well, again, there are unmet needs. We look at resolution. People want to go to more than just cellular. They want to go to subcellular, want to attribute characteristics to organelles. Don't take no for an answer anymore. Actually, when you say, like, I cannot detect that, I cannot visualize that. And at Biotechnique, we try to do that, like for instance with our microRNA scope, where we go as low as 17 nucleotides of detection. So that means that you can actually go to the antisense oligo level, siRNA level, but also microRNA detections together, even in combinations with our regular mRNA targets. So
Together?
Yeah.
Oh, wow.
On one side, and potentially in the future we will have an increased flexibility on that, because of course the gene therapy and cell therapy field is not standing still. So They will use an ASO together with an siRNA, and again, we will be able to show these things on the same slide together with protein co-detection.
That's fantastic. How do you deal with the noise?
Well, CO2 noise, it's not something we have overly problems with because of our patented amplification system, which is at this moment still one of the top sellers within the field, because people have always problems with that. And our state-of-the-art probe design that is done by our probe design team that we have internally actually takes care of that potential cross reactions that you can have.
Okay, so you provide all of that support.
We do an end-to-end solution in our US CRO, but we can also provide end-to-end solutions in a kit form where we also aid people in CGT with support that they need, also with, let's say, having some very special samples.
Yeah.
So, yeah.
Okay, and you provide support outside of the US, like New Zealand?
Of course, no. We are a global company, so we have a strong US base, a strong EMEA base, but also Asia Pac.
Oh, that's fantastic. I mean, so you pretty much answered my questions about what you can provide to researchers in cell and gene therapy, because that's an amazing platform that you have.
And that actually most of the time is the beginning, where we have also our automated solutions for ELISA and Western blotting. We have our GMP proteins. We're actually building up the whole workflow for CGT researchers, and most of the time that can start with scope, as we say, where it opens the door. But then we notice that people are convinced of the quality of our products, and actually they want to keep, of course, building their relationship up with their projects continuing in their CGT pipeline.
That's fantastic. If you're interested in finding out more about what Matthias and his team are doing here at Biotechnique, come and see him at booth 102 at ISCT Paris 2023.
So, Ted, do you hear about the roundtables?
I've heard about the roundtables.
You know, we have 50 roundtables. I don't know if you know, but I was one of the co-chairs with Quasim and Dominic.
You all did a fantastic job. Thank you.
They're so good to work with, and we work very hard. And of course, it's not only the two of us; it's the people that gave us the ideas for the roundtables, and
it shows.
Yeah, it shows. It was amazing.
Yeah.
So,
and speaking of roundtables, I had the opportunity to interview Laritis Ikonov, who is the chair of the ethics committee with cell and gene therapy.
Okay.
So he participated in some of the roundtables as well. So let's listen to that interview.
Okay, let's do it.
Bonjour. Back on day 2 with Dr. Ikonov. We are here to talk about some of the aspects of the annual meeting and how we design and frame the annual meeting and the components that you engaged with. How has it been participating in the planning, and what special sessions have you been engaged in creating?
Yes, so of course it's a great meeting. It's always good to be back in a city meeting, especially in Paris, a city that I really like. So I've been involved in both the planning and also the participation of several roundtables. So I was a moderator for 2 roundtables on hospital exception, which is a European Union provision to offer ATMPs, advanced therapy medicinal products, in a limited scale. So that's a hot topic, contested topic. So we had planned 2 roundtables, 2 hours. Full hours to discuss this topic yesterday. It remains contested. We had a lot of interviews, a lot of
comments, a lot of people
strong views and contrasting views. But I think that's a great. It was a great forum for public deliberation. And I've been also a panelist in a roundtable on public trust, which is in research in science post COVID-19, which is a very interesting topic. So how we do to restore faith in science and research, how people can trust that what scientists and researchers say is good and valid, and they can follow guidelines and recommendations. And tomorrow we have also a very interesting roundtable in the morning. I'm a participant in about how, in fact, clinics that offer unproven cell-based products, claimed MSC products, mesenchymal stromal cell products, affect legitimate research and development in the field. How can address this issue in between us and also in collaboration with regulators.
Okay. So you had a contested session. You've had hopefully not as a contested session, though I do enjoy the contested sessions.
Yes.
A lot of opinions come out, but a lot of ideas are generated from those as well. So
Yes.
wonderful selection, wonderful additions to the whole conference in the meeting. So thank you so
and
much.
with a wide
Really
array of
appreciate it.
subjects. I like to participate to a lot of things, talk about the different things. That's something I really enjoy.
All right. Well, fantastic. It's been wonderful speaking with you. I always enjoy meeting the leadership within ISCT. It gives me hope for the future of the whole community, as with the ESPs that I've met as well. So,
Okay, thanks so much.
thank you.
That's great.
Take care.
Alicia spoke with Jenny Steinberg about the Q self-processing system. Let's hear a little bit more about the system. Over to you, Alicia.
We're here with Jenny from ScaleReady. So ScaleReady are the kind providers of our Wi-Fi. So Jenny is the commercial director of ScaleReady. Would you like to tell us a little bit about your company?
Yes, I'm happy to. So ScaleReady is a joint venture partnership that was founded 2.5 years ago by 3 powerhouse players in this industry, with Biotechnique, Fresenius Kabi, and Wilson Wolf, all bringing gold standard tools and technology to the industry, tools that today are used for commercial drug manufacturing of CAR T cells.
So I'm really interested to learn about your machine Q. So that was just on the market last year, is that right? Yeah?
Yeah, it came out on the market in October last year as the final version of it. It's now been adopted in a wide variety of companies and institutions for fill and finish and freezing of cells.
Do you want to tell us a little bit about what makes this machine unique in this space?
Yes. Q is a small-scale cell processing device that is washing the cells, that is formulating the cells before it's filled and finished. The nice part with Q, it's bringing flexibility to the market. So it's not an all-in-one solution that it's always doing the same thing. We will adopt it to, all the clients' needs. So some may want to wash the cells, some may want to formulate the cells, some may want to go all the way and do all the things. Q can also formulate into different containers, so vials, bags. In the same run, it can formulate into small and large volumes, so that is unique.
What's the lowest volume it can go down, I think?
Yeah, so usually we go down to 2 ml as the lowest. That can, of course, be tweaked, but
which is amazing, right?
It's amazing. It's a perfect volume for a QC sample, and then in addition to have a product that maybe is 50 or 75 ml for the actual material that will go into patients later on.
Yeah, that's really fantastic. I feel like you've really listened to what the market's been saying, because I think we've been saying for a long time, please can we have small volumes?
Yes.
Okay, so do you want to tell me just a little bit about how this fits into other CGT platforms, in particular the G-Rex?
Yes. So with the Q device, we can wash and formulate cells both upstream and downstream of the G-Rex. So the G-Rex is our cornerstone and really the gold standard method to grow cells for cell therapy manufacturing. So Q can either be used to wash the apheresis material, or it can be used after harvesting the G-Rex for the final washing and formulation. So really at both ends. And this is where our application team comes in really handy. So what sets ScaleReady apart is the power that we have with our support organization. So the entire ScaleReady team is consisting of process optimization experts. And they will work with every client very closely to ensure that their needs are being met, utilizing the tools and technologies that we have.
So whereabouts are you guys based? Are you in the US or are you global?
So our headquarters is in the US. However, on the European side, we have a fairly big team that is operating virtually throughout Europe. So the nice part is that we have team members in different countries talking also local languages. So we are supporting both the European and the US market.
That's fantastic.
Yes.
So we'll wrap it up now, but thank you so much for coming and talking to us. And if you want to talk more to Jenny about her amazing queue here, come and see her and her team at booth 84. Thanks.
Isn't Alicia great?
She is wonderful.
It's been wonderful working with her. But ISCT's focus this year, or ISCT's focus generally, is regulatory bodies and regulatory approval of cell and gene therapies, correct? Advanced therapies. So they're focused on EU and SOHO. And I believe that you attended the SOHO roundtable?
It was amazing. It was packed with people, and it was very informative about what is coming up with the SOHO. So, I learned a lot. It was very informative.
Fantastic. Well, we have another interview with President-elect Miguel Forte. Alicia's taking care of that, so let's pass it off to her.
Okay.
Bonjour. We are here on day 2 of ISCT TV. I'm here with Miguel Forte, our incoming ISCT president. And we're just going to do a quick recap on the roundtable sessions yesterday. And in particular, we're going to talk about the hospital exemptions. Now, Miguel, you have some concerns about how the hospital exemptions are being used within the EU. Would you like to tell us a little bit about that?
Cool. Thank you for bringing us back to that discussion yesterday and pointing out to the topic, hospital exemption. Let me just, before going on to the concerns and what hospital exemption represents, highlight the fact that there was a great opportunity that ISCT is vested in at the meeting and beyond the meeting of bringing the different stakeholders to discuss topics that are relevant for patient access. Because in the end, what we're talking about is patients getting access to cell and gene therapy products. Now, hospital exemption is something that was established when the ATMP legislation was created as a bit of a steam valve for some things that couldn't fit, but has been now used by the member states as a diverted access, nearly as a parallel regulatory process. And that is a concern that was highlighted there, and I share, which is diverse use, harmonized use across Europe, and in fact, with the risk of creating a parallel regulatory track. Everybody agreed there that we should have registry, we should have transparency, we should learn about it. Now, hospital exemption has a role and should keep that role, which is access for products that have a regulatory assessment but are still not on the market. I think everybody also agreed that commercialized products, centralized, should not be put in competition with products through the hospital exemption. So bottom line, the new legislation that the European Union is forthcoming for, it will bring some rationalization into it. And I think that's where these concerns are important to be brought up to the Commission so that the legislation reflects what the field is thinking about.
Yeah, I really liked how you described it as something to fill the void where there are no other pathways, but it should be an exception, not the rule of commercialization. So you talked a little bit about some improving commercial competition within the EU. What sort of steps do you think need to be taken to do that? Because that's a big ask. But
yeah.
What are your thoughts on that?
My main concern is to ensure that today, tomorrow, and in the long run, patients in Europe have access to innovation and to products. We're seeing some alarming signals. The more alarming signal has been some products that have been put on the market after regulatory approval, engaged on reimbursement discussions which were not successful, and then the companies took the strategic decision to withdraw them from the market while they remain on the market in the US. Patients are patients across the globe on both sides of the Atlantic. So that's a concern. Other indexes like the number of clinical trials, the number of new companies, the level of investment, all of those levels are trailing behind the US. Let's not even talk about Asia where they're exploding, but that's a bit of an artificial context. What we need to do is really just analyze that and try to improve that competition. Now, we have fantastic academic research. We have centers that are top of the state of the art on science, but we need to make sure that there is support for that translation. That's kind of on the front end of filling up the innovation. Then we need to make sure that we have elements to support company creation, development of the products, and very importantly, for the patient's sake, for the innovation's sake, for the investment's sake, we need to have access. Now, we need to find a solution to making these products affordable, to making these products payable, to making these products at the right element of the choice between the cost opportunity on the different decisions that the payers have to do. But we need to tackle that on. We need to improve that. So take the research, translate the research, and ISCT is really front and center on translational path. Translate it into products and make sure that those products have the right access to the patients in need.
Do you think within the EU there's a place for state commercial partnership then, with that in mind?
Absolutely. I mean, the point I made is this product needs to be developed correctly for the sake of the products and above all for the sake of the patients. They need to be invested. Where the investment comes from, there may be different opportunities. And potentially for rare diseases, smaller ones, we may have some more, I would say, institutional funding. And for others, commercial. We need to make it flexible and support the development.
And then just one final question. What do you think needs to happen then to make these therapies available across all the countries in the EU, not just the top performing countries, but all of them?
Well, there's several things. First, there must be a centralized regulatory approval, right, which makes it pan-European. Second, I'm quite supportive of the health technology assessment centralized approach, which would mean the value assessment of these products will be valid and should be represented for the entire European country. Then there is one very challenging element, which is the budget to pay for these products, because it is member state prerogative to decide on the budget. What we think we need to do is find paths and alternatives to make sure that we equalize that access. We have strategies for equalizing agriculture. Shouldn't we have strategies to equalize medicinal product access?
Yeah. Thank you so much for your time. I really appreciate it. And it's such an interesting discussion. We could go all day.
We could go, well, we can't,
we'll
right?
wrap it up now. Thanks for joining us.
All right. I had a lot of interviews, believe it or not. My next interview was with Curate Biosciences, one of the booths here at the exhibit hall. They're dealing with cell separation in a different way than what I'm accustomed to. They're using microfluidics, which I think is very interesting. And they're going to give us some details on what their device does and how it can help.
Okay, perfect.
Sound good?
Yes, let's see it.
Let's go. Bonjour. It's Ted here again with ISCT TV Day 2. I'm here at Curate Biosciences booth 213 with Jarav Vij and Tony Ward to talk about the Curate Bioscience new device. It has some proprietary technology, DCS?
DCS, yes.
DCS, can you talk about that a little bit?
So DCS is deterministic cell separation. And so this is the world's fastest microfluidics. It's able to separate cells in a unique way as compared to most centrifugal ways. And so by eliminating the centrifugal process and integrating a very efficient 3-log wash at the same time as we do cell separation, we're achieving phenomenal biology. And so it's really addressing a lot of the industry issues around recoveries, variability, and the downstream benefits of adverse effects actually of what the platelet biology does to cells. And so, yeah, to expand on that, we're addressing regulatory T cells, we're getting better transduction efficiencies, the cells are expanding better. All in all, it gives us a shorter time to make drug, basically.
And the viability is good, right?
The viability is fantastic, yeah.
Excellent. So I heard a couple of things that were unique in what you said. So the microfluidics, I don't hear a lot about that in cell separation systems. Can you talk about that within your instrument and what microfluidics does different than what we hear?
So microfluidics is different in that it's a millisecond based separation. So we're passing cells through this array of microposts and the cells separate and get washed within a millisecond. And so that's a fundamental difference as opposed to co-locating the cells on a density gradient, for example, where we're experiencing exposure to platelets or cytokines or growth factors. So we eliminate all of those biological active agents from happening, and it allows the cells to be left gently processed without any biological side effects, which is what the industry is frankly dealing with right now, but they don't realize it.
Without a doubt. And a lot of science obviously went into this. The scientific development of the device and the partnerships that you have, can you speak a little bit about that and what the feedback you've gotten from them is?
Yeah, we have a very impressive data. I will hit on some of the key messages here. The one is it's very simple and easy to use. It's a closed system, which people love. Our partners love it. If you look at the pure performance metrics, one being the pure recoveries, we recover more than 90% of the white blood cells. And when you talk about the purity aspects, we can remove 99% of the platelets and more than 90% of the RBCs. And when you look at our end product, it's pure white versus some of the other products are pinkish and red. That really makes the day for us when people use our machine. They get the end product, which is white looking. People just like, wow, this is amazing.
Yeah, c'est fantastique.
Fantastic.
Right?
Yes.
And like I said, you have several partnerships. You've worked with academic industry. Please share all your partnerships.
Last year and this year, we have been, I think, very busy working with academia, small biotechs, large biopharma companies, and CDMOs. We have generated data with more than 10 institutes/companies. And right now we're in selling mode. We launched the product in the US 6 months ago. And the small sales team we have is very busy talking to the customers.
It's always good here.
Showing them how we can make the difference in cell therapy manufacturing processes.
Okay. And one of the things that I picked up also is, less components go in, so the product that comes out faster, comes out with less waste and less cost, and that provides access, or that improves the access capabilities for cell and gene therapy for everybody, which is what ISCT's goal is.
Yeah, definitely. I think if you put the customer hat, right, so they are looking for a consistent, easy to use product. And then everybody's thinking about cost of goods. When you walk around the floor here at the conference, there are two top of mind things. How can we reduce the cost? How can we reduce the vein-to-vein time for the patients? And we hit on all these metrics. And the reason is what Tony was mentioning. Our cells are healthy, they are better. Because of that, the transduction efficiencies will be better. So you can make the drug faster. So maybe in 3 to 5 days versus 9 to 10, or maybe in some cases 20 days.
And that factor log's less expensive when it comes down to it. So fantastic. I think you have a lovely device. Feel free to come by the booth and grab some of the scientific literature. Again, that's booth 213 with Curate Biosciences. I've got Gaurav here and Tony, so come by and say hello.
Thank you.
Thank you.
Take care. You got this.
So that's it. That's all for today.
Day 2 is all done.
It's all done. We wrap up the last 2 days, but we have more. We have still 2 more days. We're gonna have more industry, more plenaries,
more interviews, more behind the scenes.
Behind the scenes. So are we ready for that?
Almost ready. Almost ready. One thing I got to teach you before we go.
What?
I got to teach you a little bit of French.
Oh, okay.
All
Let's
right.
see.
Repeat after me.
Okay.
Je voudrais beaucoup de vin. Put it all together. Je voudrais beaucoup de vin. We want a lot of wine.
Yes. See ya.
Take care. Au revoir.
Au revoir.